Yeah, and if also I would like to just mention, yeah, I think somehow on this screen I don't want to put everybody's faces up because then I don't see my slides, but please unmute yourself and ask me questions whenever anything comes up. And I'll also make sure to leave time at the end for discussion as well. Okay, and so after discussing generally what epigenetics is, then I'll talk about what we are doing in our lab, where we use rodent model mice to ask this question about epigenetic inheritance, and finally kind of leave you with where the field is in terms of humans and whether epigenetic inheritance takes place in humans or not. Okay, so let's get started. So we all know that DNA is the genetic material and it's present in the nucleus of the cell. And DNA, and you also learn from Barry's lecture, the central dogma of molecular biology, that you go from DNA to make RNA, from RNA to protein. And the genes which are present, which make these different proteins, are present on the DNA, right? So DNA is not this one single long molecule in the nucleus, but it's present at what is known as chromosomes. So chromosome is a long double-stranded DNA molecule. So it's about 150 million base pairs long. Remember all that ATGCs, that genetic code that we have, that's present in our cells in form of these 46 chromosomes. So 46, and I will get into that in a couple of slides, is basically 23 pairs of chromosomes we have. And each of this chromosome then has these thousands of genes on it. An individual gene is about 8,500 base pair long. So you can imagine if this is the DNA going in, there might be a gene here, another gene here, then another gene here. And there's, of course, all this other DNA sequence, which some of it, we know the function of it, and some we don't know yet. So the other reminder is that all cells in our body have the exact same DNA. And this is something which, as I think early in my school as a biology student, I was very fascinated by this thing, that we all come from a single cell. So when fertilization takes place, it's a single cell formed, which keeps dividing to make the embryo and to make the whole human body. So we all start with a single cell, and so all cells in our body have the exact same DNA sequence, the same genetic code. However, in our body, we have around 200 different cell types. You have the muscle cells, the brain cells, the skin cells, heart cells, and so on and so forth. So how are these different cell types made is explained by epigenetics. But before I go, I kind of keep reminding everyone the central dogma and what I mean by the terms that I use. So when we go from DNA, where the genes are, to make RNA and then to make a protein, we call this as gene expression. So the gene is being expressed, it's making its product. Okay, and this is something which I will use during my lecture today. So I kind of wanted to bring everyone on the same page. So when I say chromosomes is what our set of genetic information is, what does that mean? So this is a set of chromosomes in one of the cells in your body, okay? We have, and in each, those, they are present in pairs. So we have 22 somatic chromosomes, and then 2 of these are sex chromosomes. So one pair is a sex chromosome, okay? So these, when we say this is one pair, is chromosome 1, chromosome 2, chromosome 3, we have 2 copies of each chromosome. One we get from our mom, one we get from our dad. And each pair is basically has exact same genes on it. They have something known as different alleles, but we're not going to get into that today. So the genes which are present on chromosome 1 are different from genes which are present on chromosome 2. So each chromosome carries its own genes. There are thousands of genes on one, and total we have so far identified with the human genome sequencing that there are about 20,000 to 25,000 genes. These two are the sex chromosomes. So if you have two of the X chromosomes, you're a female. If you have a 1X and a 1Y, then you are a male. And so this is how every cell of your body would be. This is the genetic information. Now, how do we pass down this information to the next generation? That happens by what is known as germ cells, which would be sperm in the case of males, and then egg in the case of females. So this is actually an image of an egg being fertilized by sperm. And this is not the actual color, it's been stained to look this way. It's not pink, the sperm is not pink. But what you can see here and appreciate is that egg is very much is at magnitude of 1,000-fold bigger than a sperm. And that's because the egg supplies a large amount of additional information for the early embryo to grow. So mother supplies many different factors, the bag of goodies, you can say, which is required for early development. And sperm delivers that set of chromosomes, which from dad we need. So for all these different sperm, only one is going to fertilize. And then once it's fertilized the egg, it's going to send a signal to all the other sperm will be then blocked. So this is the process of fertilization. Now, what happens at the genetic level during this time is you have, this is the egg and this is the sperm. So our germ cells undergo a special kind of division so that when they're developing, we actually reduce the number of chromosomes. So in every cell of our body, the somatic cells are muscle, brain, heart cells. We have 2 sets of each chromosome, but in our germ cells, we have only 1 set. And that's because we don't want our babies to keep accumulating, right? So in this case, the egg has 1 of each chromosome. The sperm has 1 of each chromosome. Chromosome 1 here from mom, chromosome 1 here from dad, and so on and so forth. When they fertilize, they both give their genetic information, and the baby gets their set of 46 chromosomes, 23 pairs. And in this case, this is a girl. There are 2 X's here. Okay, so this is how genetic inheritance takes place. We pass down our genetic information, and that's the major form of inheritance. That's how our species is progressing. We maintain our genetic information so we can keep producing more humans and keep everything the same. Now comes into play epigenetics. Okay, so what is epigenetics? So genetics is, as I described and you've learned already, is that it controls our appearance, our behavior, our health by the genetic code, the DNA sequence, which has all these different genes, which will make the proteins, which will go on and do all the different functions in the cell. Epigenetics is the process of regulating this appearance on behavior and health by controlling how the genetic information is decoded. So this takes me back to when I said that we have 200 different cell types in our body, but they're all genetically identical, but very different in their function and structure. So for instance, a skin cell is very different from a brain cell. Although they have exact same DNA sequence, a cell is doing very different function, looks very different than a skin cell. So what is happening? How do we get that different level of this so different expression by having exact same DNA? So this happens, you can take an example of, let's say there is gene X, which is required for skin cell development. So we want this gene to make its product, that's the protein, only in the skin cell, but not in the brain cell. So by these molecular mechanisms, we can block this thing to happen in the brain cell, but let this gene make its RNA and protein in the skin cells. And this is regulated by what is known as epigenetic gene regulation. So how does that happen? How do we make this gene not make its RNA and protein in the other cells, in the brain cell, but do it in the skin cell? So the first level of this regulation comes at the level of packaging of DNA. So another quite interesting feature about our DNA is that we have this 2-meter long DNA, which is somehow packaged into this very small nucleus, which is about 5 micron big. So you can imagine it's 1,000 times smaller than the tip of a needle. And you have to somehow package. So if you open your cells and get all the DNA out and stretch it out, it's going to be 2-meter long. So how do we package that fiber into such a small space? And this is done in a very practical way. You can imagine at home, you have this charger for your phone. It has long cord. If you just try to just put it together and throw it somewhere, it's going to take a lot of space. But if you take it nicely and wrap it around the charger, it's going to become small and you can then pack it nicely. This is exactly how it's done in the cells too. So something known as these balls, which are actually 8 proteins which come together, it's called histone proteins, and this is known as nucleosome. So these beads are used, which are proteins, to use the package to package the DNA inside the nucleus. So you basically have this bead and the DNA is wrapped around 2 times, and so slowly you keep wrapping and then packaging it. And then after this beads, you further package this together, and there's a whole higher order chromatin, it's called DNA packaging, which is still a very active area of research, how that happens. And that's how you package this really long DNA into the nucleus. Now, while packaging this, what you can do is you can also regulate which genes are expressed. So which part of the DNA is accessible for it to make the protein and which part is not. So for instance, if you look at this, here you have these beads quite tightly packed. So that means factors which bind to DNA to make protein, they cannot access this. So these genes will be off. But areas where there's more loose packaging, here factors can go and bind and access DNA, and that would allow genes to make their product, the protein. So this is one way. So you can imagine maybe in a skin cell, the gene which is required for skin cell development would be in this form, but the gene which is for brain cell development would be in this form. Any questions so far? Sorry, let me know if I'm going too fast or something. Otherwise, I'll keep on going. Okay, so the other way we can do this is by modifying the DNA itself. So you learned about the bases in the DNA, ATGC. So the cytosine can be, a chemical group can be bound to this base, which can then make the DNA, again, less accessible to all those factors that bind to DNA to make protein from these genes. So if it is modified, so this is known as, methyl group. If it is modified, then you don't express that gene, so the gene is off. If it is not modified, then the gene is on. Okay, so that's number 2 mechanism. And the third mechanism is by RNA. So Barry already covered this in his last lecture, where we learned that not all RNA make protein. So you go from DNA to RNA to protein, but some RNA are called the non-coding RNA. So they don't make protein, but they carry out other regulatory functions in the cell. Some of these are known as small RNAs just because they're small. So they are just 18 nucleotide to 25 nucleotide long of that ATGC stretch. 80, not AT, but U. So RNA has one base different. And so these small RNAs can go and bind to mRNA, the RNA that makes the protein, and chop it. And then thus it prevents this RNA to make the protein. So this is another way where maybe in the skin cell, it started making the RNA for the brain cell development, but the cell checked, well, we don't want this protein to be made. This has to be made in the brain cell. So then these small RNAs go and chop this RNA so that it cannot make the protein. So that's the third level where you can regulate which genes would be expressing or not. Okay, so there are these 3 major ways. There are some other processes, some other factors which can regulate gene expression as well. But epigenetically regulation of gene expression is by these three major mechanisms. So it's great that people were talking about symphony today and orchestra, because that's the analogy I wanted to discuss. So I know what all I discussed could be kind of confusing and hard to understand. So I thought I'll give this analogy, which is quite often used to explain what epigenetics is. And this was also done nicely in this article that I reference here. So you can imagine that all the genes are the different musicians in an orchestra. And they work together to produce a symphony. Now, one song is very different from the other song. Even though it's the same musicians using the same instruments, but they make a symphony, one is very different from the other, and that's all based on how the music, the sheet music is read, which instruments are playing at one point and which instruments are not playing at the other point, right? So to make one symphony, you might say, have the conductor will say, switch on, go drums and the piano should go on, and the violin and guitar should not go on. Then you change the note, and now the piano has to go higher, and then the guitar comes in and drum has to be off. So while it's all the same musicians are sitting and playing together, it's this whole orchestra is working together to make a nice symphony. So now think of this in terms of cell and epigenetics. So these musicians you can think about are these genes, which are all there in every cell, but in one cell, they will be playing a different symphony and another one, a different one, by deciding which music instrument to play at which time. So in this way, you can, using exact same number of genes, come out with different phenotype. What is written here, phenotype, is basically a trait or an outcome of that combination of gene expression that we're looking at in the cell. So in all of this, then what is the epigenetic machinery? These three factors that we discussed, those would be the conductor here, which is making sure that everything is going fine. So in a skin cell, it will make sure only these genes are on and the others are off. And in the brain cell, it will make sure that the brain cell specific genes are on and skin cell specific genes are off. And that's how you come up with a good symphony. Now you can imagine if every musician starts to play on their own and don't follow the sheet music, what's going to happen? Nobody's going to like that music, right? Same can happen to the cells, that every gene starts to express or make protein when it is not needed. And so therefore, it's a very nice balance of all these different genes and when to express and which ones to express, which make a nice phenotype. In case of music, they make a nice symphony. Now, you might say, well, what if we remove some of these musicians? What would that mean? Well, removing the musicians, of course, the band will not be happy. Secondly, that would mean you're actually making a change in the DNA itself, right? Because then, let's say in the next symphony, in the next song you're playing, you want the drum to come back, but now you've already asked the drummer to go home. Now you cannot play that. So the good thing about epigenetics is that everything is there. It's there in the DNA. You're just using it at different times. And so that allows you that dynamic of allowing different gene expression while keeping our genetic code the same. So I thought I don't know if I'll be able to play it, but I had a nice video which kind of describes this. It was done by Nature Journal. Let me see if I'll have to stop sharing. And in the meantime, think about, I know I provide a lot of information. So I'll stop sharing this and I'll try to share this video. Yeah.
Lumina.
If you change the notes of Beethoven, you'd probably get a strange reaction. But you don't have to change a music score to change how it's expressed. And it's the same in our genes. You don't have to change the DNA itself to change how genes are expressed. Almost every cell in your body has the same DNA sequence. But of course, not all cells are the same. That's because different cells use the code in different ways. Stuff happens to the genes during development and through life. They're turned on and off depending on their jobs. A cell's combined set of all these changes is called its epigenome. The term epigenome describes changes that affect gene expression that aren't changes to the DNA itself, the sheet music. Epigenomic changes ensure that a heart cell is different to a brain cell, even though the code is the same. New work is helping to provide a reference of the epigenomic landscape in lots of different human cells.
Okay. I mean, I could have played this. It's a long video, so I thought I'll just leave it here. But this kind of gives you an idea, and I can also send Barry the link if you guys want to see the whole video. Basically, that's about it with the introduction about this. Any questions so far? I'll just share my screen again.
Yes. Let's see here. Wait a minute. I have a question. Yeah.
Mm-hmm.
Hello?
Go ahead, Constantine.
Oh, so we found out how these things change, how the system of epigenetics change. Do we know what the influences are that make them change, or that's the next part of your lecture?
Yes. Yeah. So, I mean, to some extent, so it's a very good question as to, okay, we have all these, epigenetic mechanism which are making a skin cell, say a skin cell. How do they change? And I would say it's still a very active area of research, both at the level of a single cell, like how it develops and makes one cell type versus the other, and also at the organismal level. And that's something we're trying to understand. Others are too. We have some information of, for instance, environment can actually change these epigenetic marks, and as what I will talk about. But yeah, it's an active area of research. We still have many questions unanswered. But yeah, some of it I will talk about in the next part of the talk. So if we have more questions pertaining to that, we'll get back to that towards the end.
Thank you.
Mm-hmm. Okay, so that's a question, like how does this epigenetic changes that we saw, do they affect human health? So we have seen that at the level of a cell during development, how that affects which cells are made. In terms of, more of a health perspective, individuals differing from one another epigenetically, there is very good evidence from twin studies that epigenetics plays an important role in our health as well. And this would be, this was kind of, you might have seen around you as well when you look at some identical twins, which are genetically identical. So there are twins, which these identical twins come from that single cell. So when mom and dad provide their genetic material, you make the baby cell. That baby cell then, which has that set of chromosomes, that divides to make 2 embryos instead of 1. So they start with the exact same genetic material. But we have seen many a times that twins, as they grow, develop different traits sometimes they develop different diseases. So then the question is, if everything is dependent on your DNA, then that doesn't explain what happens in twins. So twins are a great model, you can say, to study what happens, what is epigenetic and what is genetic. So what one study showed was very interesting was that when twins start off early, when they're born, they're of course very similar because they have the same epigenetic information, epigenome. But as they grow older, they found that sometimes they can differ, and one twin can develop some diseases which the other one doesn't. And a lot of times it's due to the epigenetic changes they have accumulated during their lifetime. So this is quite interesting and tells us that how epigenetics can play a role in our health as well, and also suggests a very intriguing hypothesis, a possibility, which is, can we transmit all these changes we have acquired during our life in our epigenome to our next generation? So the model would be that as we are, growing, we experience different things in our life, our environment, be it good things or bad things, that affects our epigenome, as we learned all the epigenetic information molecules in our cells. And then we pass it on to the next generation, which can then affect their health and lifespan. So in case of females, it will be through eggs, and in males, it would be through sperm. So the first question is, that sounds like an intriguing possibility. Do we have any evidence that this happens? So indeed, there is some evidence from human studies. And this was kind of where this field kind of restarted, I would say, the whole idea that this could happen. So this study showed that ancestral diet can affect metabolic phenotypes in descendants. So this was done, this is known as overcalyx cohort study. This was done in 3 different generations of families in a province in Sweden known as overcalyx. And what they found was that, so basically what they did, these are called epidemiological studies. What these do, they, of course, on humans, we cannot conduct experiments for obvious reasons. So we collect data, as much as data is possible around available, and then make our interpretations based on that. So what they did in this study during late 1800s and early 1900s, they collected information on all the public records of which year there was good crop season, so they have enough for food, which year was not a good crop season, and there was less availability of sufficient food. Then they also collected public records of the health, so deaths in the families and the reasons for those deaths. So by using all that data, what they found was that the grandfather's food supply was linked to mortality rates in grandsons. So in other words, if there's plenty of food during grandfather's growing years, then the grandson was more likely to die from metabolic diseases such as obesity, diabetes, and cardiovascular disorders. So this was quite a striking study and kind of pointed to that indeed these, something that our environment, the environment we're exposed to, is affecting our future generations' health. Now, this is considered to be via epigenetic mechanisms because, as I said, our DNA doesn't change so quickly. It takes millions of years of evolution to make changes. So this is most likely via epigenetic changes because epigenome is dynamic. It can be changed. Genes can be turned on and off, right? We cannot delete genes, but we can turn them on and off. So we can make changes to the epigenome. So while this was very exciting, it's still a correlation, right? So it's a link. They saw this, how much food was there and what's happening. So that's one of the big limitations for any study we do with data from humans, that it's mostly correlation based. And we cannot actually study mechanisms. Can you imagine doing experiments on humans, putting them in different diets and seeing what happens to their grandsons? That would be A, too much time to do that experiment, B, ethically not right. So that's when we in labs think about other ways we can ask these questions. And what we do is we use model organisms to answer these questions. So if this is happening, if we're passing down this information to the next generation, then whichever environment we're exposed to should affect our body somehow, our somatic cells, our brain cells, our skin cells, our lung, our heart. That information then has to be passed down to our germ cells. And since the overcalyx cohort study, the first one was on the paternal line, so I'm using paternal line here to show this. So this information has to be then passed down to our germ cells, the sperm, passed to the egg at the time of fertilization, when that sperm and egg meet. Then it has to, that information, that epigenetic information or the information of the environment has to then undergo all these early developmental changes, be maintained to finally have this outcome in the offspring of, say, a diabetes or something else. So it's a pretty complex process. And that's why it's more challenging to test this, and many have challenged that this even happens. So we need very good model systems to ask this question and do the experiments in a controlled manner. So how do we study this? And this is known as transgenerational epigenetic inheritance, because we are passing to multiple generations. So in search of experiment models. So first question, when we think about asking this question in the lab, that this is actually happening or not, and if we can pass down information about our environment to our offspring, we need to know which organism to study. And throughout, there are different labs are studying different model organisms. But one thing we want to do is to use something known as isogenic population of animals. So we want the animals that we're conducting these experiments on to be all genetically identical. Otherwise, we will not know what's genetic and what is epigenetic. And this is, for instance, in our human population, we all have variants of our genes, some known as alleles. So we are not all same genetically. We are 99% same, but there's some changes between each one of us. But in terms of doing experiment, we want to make sure that the animals are all genetically the same. So we start with what is known as isogenic inbred mice. So these mice are inbred, they're not outbred, so they are very low chance of them undergoing any genetic changes. We use mice because these are mammals, these are vertebrates, so they are closer to studying human biology. The common house mice is, musculus is very well characterized. We know the genome, we know how many chromosomes or what genes they have, and all of it is very well worked out. So it is easy to work because of that too. Their lifespan is, you produce offspring in about 21 days. So it's not the same, it's not as short as maybe studying using some other organisms, but still it's short enough compared to maybe using a monkey or a primate model. Okay, so one thing is having this. There was a question, so I'll just go ahead and read that. How do you remove confounding effects other than grandfather's diet? What is the grandmother's diet? What about selective effects in the wound? What a significant fraction of fetuses not surviving? Okay, all great questions, yeah. So and that's another important point as to why we need to do this, an experiment. So this was a question from Mark, as to why we need to do these experiments in laboratory, where we can actually look into all these other confounding factors. And that's why, although that's a very fascinating study, gives us motivation to test this, the human study has so many other confounding factors. We are looking into, and the one part of the question, which I'll kind of talk about now itself, but about grandmother's diet. And so, yeah, so when you're making a baby, you're getting your genetic and epigenetic information from both your dad and mom. And we don't know what's coming from where. There could be a combination of effects. They could be even canceling each other. Who knows what goes on? So right now, I would say experimentally, the field is in its infancy, in the sense we're only trying to understand one side of inheritance at this point. But the next step in the field would be to see what we inherit from mother, what we inherit from father, and how the two interact. So right now in the labs, we're going to focus on one side, but there's going to be this from both ends. We're going to have, we will be inheriting epigenetic information. Mothers definitely have a much bigger role to play, because the fetus develops inside the womb. And so the fetus is directly exposed to the environment the mother is exposed to. So I did not talk about, I'm just checking the time where I am. So I did not talk about this study, which was done on the female germline, where they looked at kids who were in utero at the time of World War II, towards the end of World War II, when women were getting less than 1,000 calories per day. So they were getting very low amounts of calories. The kids who were growing in the fetus at that time, when they grew up, they had metabolic disorders. So that suggests that something during this early development affected their epigenome, that they had these disorders when they were grown-ups. Now, in that case, we don't know if it was something coming preconception from epigenetic change in the egg, or just what the mother was eating at that time and a direct interaction of the fetus with the environment. So it's, in those studies, it's hard to separate the two out. And that's a major reason why most of the labs right now are studying paternal inheritance, because it's easy. Male would be mostly just providing the sperm, and that's so small, and it's mostly the genetic information, and a lot of it comes from the egg and also the long-term interaction of the fetus with the mother. Okay, so we can discuss this more towards the end, but I'll go to the next step now. Okay, so one is identifying which model. So we started with mice, which are inbred. Second is, now if we want to study this in our lab, this mechanism of how this information is going, we want to first have clear defined phenotypes, what we saw in the humans. So to do that, we set up these experiments in our labs, and we do different, you can study different paradigms. Our lab studies effects of different diets, so a low protein diet. How we do these experiments is we put these mice from the time they are weaned off from their mothers onto different diets, either a controlled diet or either a controlled diet or a low protein diet. So low protein is, so in the mouse feed that we give them in the vivarium, usually it's 19% is protein, but the low protein carries 10%. So it's not a complete loss, we are reduced the amount of protein. And then we keep them on that diet for about 8 to 10 weeks. And then we take the males and mate them with females, which were always kept on a normal diet. So control here means a normal diet, a balanced normal diet. So then we let them mate, we get their pups, and then we look at their phenotype, so their metabolic health, so their gene expression changes in their pups. And what we found is that males which were fed a low protein diet, they sired offspring with altered lipid and cholesterol metabolism. So this was done in our lab, in my postdoc lab, in OLLI Rando's lab. And around the same times and now following years, many labs have looked at many different exposures, be it exposure to smoke components like nicotine, psychological stress. So you can give mice different stressors as they're growing up, or give the mother stressors when they're in the womb. Then we have also their labs which are studying exposure to different toxicants like DDT, TBT. Some of these are endocrine disruptors. They affect your endocrine system. And of course, we study low protein diet. Now other labs have looked at high fat diet, Western diet, even reduction of the calories. So over this last decade or so, basically over the many different labs throughout all over the world have looked at so many different exposures to the paternal generation, and this is all rodent studies. And what is the common outcome from all of these is that the next generation shows changes in their health. And in most cases, it's metabolic changes. And I would say it still remains an open question as to why metabolism. It could be that they just checked metabolism because it's easy to check, as opposed to some other changes which might be going on. But it's kind of, when this whole, this paper from OLLI Rando's lab came out in 2010, it was kind of the first study in rodents showing what people saw in humans, right? So this is basically what they saw, where in the Overcalyx cohort that the ancestral diet was affecting phenotypes in the progeny. So we saw this in mice. Now with all these other exposures, we see the same thing. If you expose the father to different environments, their offspring has a change in their health outcome. So I think now with all these studies, that part is very convincing, at least in the rodent model, that this happens. Question now comes as to what is the mechanism? How are we passing down this information to the next generation? And which is where all these grants are coming up, because everybody is now interested, like how are we passing down this information? So how does this work? So we start with setting up model organisms. We see if this is happening in the mice. That's check. Now is the question, how does this all work? So if you think about this, so male parent is passing this information to the offspring. What happens during this process is mating, and then you get the offspring. So during fertilization, some of this information is being passed down, and there could be many factors that affect this. This could be just maternal judgment of the male quality. So sometimes in animals, a female can judge the male quality. Maybe a low protein male is not as healthy, and so she would put less resources on her pups. This could be something even in the microbiome being transmitted, or some kind of proteins or something in the seminal fluid, or this could be in the sperm, the epigenetic information. So we wanted to test that directly, whether this actually, we are passing this information to the next generation in sperm. Is this information transmitted in sperm, and how do we do that? So what we do is known as in vitro fertilization, or IVF. So you may have heard this as an assisted reproduction mechanism. Many couples which are not able to have kids do go through this route. But this is also a very good system for us to test our mechanism in the lab. So what we can do is we can collect sperm from either a mouse which is on a controlled diet, like a normal diet, or a low protein diet, and get egg from a female which is on a normal diet, and do in vitro fertilization. So now in this case, there's no other confounding factors coming. The only contribution from each parent is their gametes. So this is where in the lab, we're able to get rid of all those confounding factors. We only take the sperm and the egg. Kids have never seen their parents in this case. They only get this information, the genetic and the epigenetic information. We get the fertilized embryo, we implant it into another mouse. It's known as a pseudo-pregnant female, the recipient female. And then we get the pups from there and then look for what happens to them, their metabolic phenotype. So what we found was that in this case, when we only use the sperm from the male and not the whole male to mate the female, we still saw those changes in the offspring that we saw in this case in the natural mating, the lipid and cholesterol changes. So this suggested that in fact, this epigenetic information is indeed carried in the sperm. So this information of the paternal diet is carried in the sperm. So now that we have narrowed it down to the sperm, the question comes, which epigenetic information carrier? So remember I talked about it could be how the DNA is packaged, or the modification on the DNA, the methyl groups, or it could be the small RNA, which can then bind to the mRNA and not make protein. So what we did is we asked this. So, okay, I'll just, I forgot the slide was there. So basically, yeah, there are different steps to this mechanism. So now we know that this exposure of different diet affects the health. We know it's in the sperm. The question is, what is the information carried in sperm? To do that, we exposed animals to low protein diet, collected their sperm, and then look for what happens to all the epigenetic molecules. So we didn't see any changes in those the beads on the DNA or on DNA methylation, the methyl groups that are added. But what we saw was an increase in the small RNAs in sperm, in the low protein sperm, not in the normal sperm. So this was exciting because that suggested that it's the small RNAs which are potentially carrying this information to the next generation. So now that we know what's in this carried in the sperm, it's the small RNAs. The next question is, how is it that we pass this information from our somatic cells to the germ cells, to sperm? And this is kind of very exciting and new area, and I'm kind of more working in the lab right now, and I will not go into all the experimental details, but in this kind of schematic, I will show you what we think is going on. It's still a model and we need more data to kind of test this model. But what happens, so this is sperm maturation, spermatogenesis takes place in the testes, and this is the same in humans. I'm showing here is the mouse. Then sperm undergoes something known as post-testicular maturation in epididymis. This is a long tubule where sperm enter after testes, spermatogenesis in the testes. This is very important organ because this is where sperm become motile. Before entering here, so until this point, they cannot swim, so they cannot reach the female tract and reach the egg. So only during their maturation in the epididymis, they learn how to swim and fertilize. So what we found is that these, so this is a tubule. So you can imagine it's a tube and then sperm are kind of swimming between there. They're not physically interacting with the cells of this tubule, but in the lumen. So what we find that the cells in this tubule have very high abundance of those small RNAs too. And what we think is happening that these cells are secreting these small vesicles known as extracellular vesicles, these small pockets which can package cargo in them and deliver to other cells. So what we think is happening is that these small RNAs are generated by these cells in this lumen and packaged into these small vesicles, which binds to sperm and delivers these small RNAs to them. So this was very interesting and exciting because so far it's always considered that there's no interaction between somatic cells or germ cells, and that was actually kind of the major challenge to any of this transgenerational inheritance as to how do we pass information which mostly affects our somatic cells, our brain cells, skin cells, to the germ cells. So this could be a potential mechanism, and we are working to understand how this happens. But I think I will leave it at this, and I'm happy to answer questions on this later. So now comes the final question. So we know there are small RNAs which change in low protein sperm when the mice are fed low protein diet. They potentially come from these somatic cells. Now the question is, when sperm delivers this to the embryo at the time of fertilization, how do they result in this metabolic phenotypes we see in the offspring? What results in that cholesterol metabolic changes? So what we think would be happening is that sperm would deliver these small RNAs to the egg when they fertilize. These small RNAs, as you may remember, can target RNA and chop them so they don't make protein. So they will change the gene expression, and that might affect the health of the offspring. So we wanted to test that. So how we do that in the lab is we can, with that IVF, as I showed you, we can make embryos, we can generate embryos. Then we can inject with a micro injection needle these small RNAs. These small RNAs we can either purify from sperm, or we can make mimics of these, synthesize them. There are companies that make oligonucleotides. So these are stretches of AUGCs. So you have a sequence, you tell the company and they make it, or we purify from sperm. And we've done actually both of these. So what we do is we inject these in the early embryo, then let the embryo grow. Then we look for these mRNAs in the embryos. What happens to them, the ones which have this small RNAs injected. So what happens is these small RNAs go and bind to some of their target RNAs and chop them. So now this cell only has these, I'm only showing 3, but we're looking at thousands of these mRNAs. But so what happens in this one now, these 2 mRNAs are gone. They're not going to make their protein, but these 3 are still there. So this early change in gene expression might affect the health and well-being of the offspring as they get older. Now, the one big caveat, still unknown, big question in the field, and are in this particular project and from other labs too, is how we go from this early change to, let's say, cholesterol or lipid metabolism changes. It's a complete big black box right now, and we don't have an answer. We're trying to understand how early changes in gene expression during the embryo development could have these effects later on. Okay, so overall, what we think is happening is that during as the germ cells are developing and we are growing, we're exposed to different environments. Some of the somatic cells, when they get exposed, they may make these different informational signals in the form of these small RNAs, send them to sperm, And then when sperm fertilize the egg, they send these molecules to the egg, which changes some early embryonic development, which could have an effect on the adult health. Many questions are open here, which we don't know answers to, how all of these different steps happen. We are just getting early clues to the potential mechanism. So with that now, I will talk, oh, I just see there's a question in the chat. And please, that will give you all to think about what I just talked as well and have any questions. So there's a question from Owen, do these metabolic changes pass on to the offspring of the offspring? So that's a very good question. What about transgenerational, right? This is intergenerational, as we call it. It's from father to kids. What about the grandkids? In humans, we saw it's going to the grandkids too. So far, we haven't done that experiment, the short answer is. And why we haven't done is because, as a scientist, you want to make the problem simpler to answer it first, then to make it more complicated. So if we start thinking about multiple generations, we won't be able to get to the answer. So what we do is we say, okay, let's first understand how we pass this information from first father to the kids. Once we know that mechanism, then we will see if they are now passing it to the next. So it's more of how we think about this. That's why we haven't. But having said that, there are some other labs which have looked at some other exposures and looked at next to next generation. And it looks like in case of mammals, max you can get to the third, sometimes maybe fourth, but very rarely. But the exposure, basically the phenotype starts to die out after the third generation or so. Now, evolutionary thinking, that makes sense, because if we are in an environment, let's say there's a scarcity of food, we want to pass that information to our kids, but maybe when the kids of our kids, but maybe after that, the environment is going to change. So we don't want to have a very long lasting information being transmitted to multiple generations. So it makes sense to me thinking about it in that terms. Now in other model organisms, so a lot of this inheritance work is done on C. elegans as well. These are small worms. And there a lot of studies have shown that some of these small RNA signaling can actually go to 4, 5, 6 generations, and it's copied beautifully. There's no change. So which makes sense, because in worms, their replication time is very fast. So they have progeny very quickly. And for them, it might be that they're still in the same environment for even 6 generations. So the short answer is, in our experiments, we haven't done it. Some have done in some other exposures, and you would see still some phenotype in the next. But it's still an open question that how much do we pass down to our grandchildren? And if we do, what is the mechanism? Because what we pass down to our kids, so let's say if the kid has a diabetic, so outcome is the kid has diabetes. Now, what they're passing to their next generation could be just because they have diabetes, and that's why they're passing it to the next generation, as opposed to a signal that came from their father. So these are some of the questions which are very complicated. We don't know have answers to yet, but the goal is, once we understand intergenerational, then we're going to get at transgenerational. Any other questions? Okay, so I'll go to what I wanted to cover. The last step of this was basically, so we have learned this from some of our studies. And so I talked about work I've done, but there are other labs also which have come to very similar conclusions, all in rodent studies. What does this say for us humans? How do we pass down epigenetic information? Do we pass down the information on our life's experiences to the next generation? And the simple answer is we really need more evidence to support this. So I think that's where I said we are still just starting to understand this in a model system. Humans are much more complicated. So we definitely need more evidence to support this happens. There is some data that it could be. So for instance, human sperm carry similar small RNAs that we see in mouse sperm. And there was this recent study which showed that human sperm, they display rapid responses to diet. So basically, if you so they conducted this study where it was a 2-week study. First week, all the participants were given, these were healthy males in their early 20 ages, and they were given normal healthy diet. And then for 1 week, they were given high sugar diet. And they saw that in their sperm, they saw these small RNAs changing very quickly, just after 1 week exposure. So looks like at least whether this is passing down something to next generation or not, these small RNAs are responsive to the environment in human sperm as well. And it looks it's very quick response as well. So I think only with future research, we will know what this means for humans. But I think the mouse studies will provide the key insights, which we can then think about some ways of testing in humans. So the big questions which remain unanswered and what we in our lab are trying to address is that how does diet regulate small RNA levels? So how is it that a low protein diet leads to these changes? And as I said, we are trying to understand how those vesicles are passing information to sperm. That is known as the soma-germline communication. What is the mechanism? And finally, what these small RNAs do when we see those early changes in gene expression, what that actually means for health of the offspring. So all these are important questions which we hope to address in coming years. And with that, I would like to thank my lab and my postdoc lab, where a majority of this work was done, Dr. OLLI Rando's lab, my current lab at UCSC, our collaborators, and all the funding agencies. Of course, NIH is funded by your tax dollars, so thank you to you as well. And I will be happy to take any further questions, open to discussions. Yeah, thank you.
Okay, all right, questions for Sharma? Okay. Well, I'll ask one then. So what do we know about these RNAs? Are these RNAs from non-coding parts of the genome, and are the genes themselves for the RNAs changed?
Yeah, so these RNAs are from the non-coding parts in the sense that they are the RNA which don't make protein. There's many different classes of these small RNAs. They all function in their own different mechanisms. One class which we are very interested in is known as tRNA fragments. So I think you must have gone through tRNAs in Barry's lecture about translation, how those RNAs are one of the non-coding RNAs, which play a very important role in making the protein. What we have learned in the last 10 years is that those RNA, they of course carry out this function in translation, but they also get chopped to make smaller RNAs known as tRNA fragments. And then these small RNAs can regulate gene expression. So it's like biology is just so interesting. Everything you learn, you find something interesting furthermore. So tRNAs were already super cool in doing what they do in translation, but now it looks like they can make further small RNAs, which are now regulating gene expression. And they are one of the really abundant small RNAs in sperm. And they change with low protein diet, with high fat diet. So that's one where we are really interested in understanding what they do. There are some other classes known as MicroRNAs, which are again these short stretches of RNA. They are made in the same way as an mRNA, using the same machinery, but they are the longer RNA is then processed, chopped to make these small RNAs, which can go and bind the target RNA and chop them. So there are different classes, and these two are the ones which we are really interested in understanding their function. I think I saw someone had their hand up earlier.
Mm-hmm.
Maybe, yeah.
Another question here?
Yeah, Ralph.
Yeah, Ralph, unmute yourself.
Well, I was wondering whether there's a sort of logic in what the small RNA is doing. Is it making a change which is designed to aid survival under those conditions that the parent has encountered?
Right. So whether there's an adaptive response here? Are they making them in any way fit? Like the question would be, why are we even sending this information to the next generation? Are they making them fitter? Why we pass down this information? And this is again where so far data looks like, for instance, from humans, it looks like it's actually not adaptive in the sense the kids end up having metabolic disorders. Now, there is an explanation, which is a hypothesis at this point. So let's say you are not getting enough food and you think your kids will not get enough food. So you pass them that information. But now your kids have plenty of food. So your body still thinks that, oh, we have to conserve all this fat because I'm not going to get food, but you're getting enough food. So in that sense, it becomes maladaptive. So you're basically sending the wrong signal. Now, if you put your kids in a low, there's not enough food, then they will do well. And that's the prediction. So for instance, we haven't put our mice, these low protein mice, they're pups on low protein diet. That would be one experiment to do at some point, but maybe they do better on a low protein diet because otherwise you're making them sicker by sending this information. So this is where somewhere it's looks like it can be of advantage, but only if the kids are in the same environment. And always kids are going to interact with their environment too. So I will talk about one interesting study which actually provides an adaptive response. So in same lab, or Dr. OLLI Rando's lab, they're also studying exposure to nicotine. So nicotine is one of the major products in the smoke. And they wanted to know if that information is passed on to the next generation, how that affects kids. So what they found is that the kids of, and this is whenever I talk about it, I say, okay, I'm not saying you should smoke, but the kids actually are better adaptive at taking higher doses of nicotine.
Mm.
So they do better with higher doses. They survive better at higher LD50 doses. What was more interesting was not only nicotine to cocaine as well. So what was found is that their liver has undergone this gene expression change, which can clear out these toxicants better than the kids whose fathers were not exposed. But this was only, phenotype was only exposed when they exposed the kids to nicotine or to cocaine. So if you look at just normal kids, then you don't see any adaptive response. So only when they were challenged with the nicotine, then they saw that they actually now have a better survival. So there seems to be some adaptive response there, but yeah, it's still an open question. How much? The other important question is how much information do we send? Do we send an information more broadly that, okay, I didn't have a good life, my sperm is not good, here you go, or it's like, okay, I was getting this much of water, but the air quality wasn't good, and I was stressed. So is it more of like a digital information we're sending, these all different things, or more of general quality of life? So there's ways to address that, which some labs are trying to do, is where you expose animals to many different conditions and then see how their pups behave. Is it that all of them have the same effect? They all have glucose metabolism defect, or if the father was exposed to a different toxicant, they have different effect, and if a father was exposed to a different diet, they have a different effect. So those are some of the questions which would be important to know what we're passing down, right? It could be very cool if we're actually sending very specific information, or it could be just we're making not a healthy gamete, not a healthy sperm, and that's what affects the next generation. And those are all the questions we need to kind of do much more experiments to actually support either of those hypotheses.
Okay, other questions for Upasna? Okay, well, I'll ask another one then. So just a kind of a technical one. So after you do the experiment, you have the progeny mice and you test them to see what genes are turned on or off. What do you actually test? I mean, do you take a piece of the liver or just a blood sample, or what do you test in the mouse?
Yeah, so the original study from 2010, they looked at both the blood and the liver samples. So liver was generally, you kind of, when you think about diet and metabolism, you go after liver. So they saw gene expression changes in the liver, and in the blood, they looked at lipid cholesterol levels. And that's something where I think the field is limited. Most labs do this. It's easier on mice to collect their blood and to collect some of these tissues, as opposed to maybe doing like an intensive behavioral tests or multi-organ tests. So some of these we might be actually losing that information because we're not testing the next generation for 1,000 different parameters, but maybe a few of them. But yeah, so the original which was done was gene expression changes in liver and then blood samples. Now we're kind of, because we know that this is being passed down, now a lot of work we do in the lab is all about the paternal generation, what's going on in the sperm, how are we changing these RNA, what these RNAs do during early development. So we're asking some of those questions now.
So one thing it might be useful to tell people, Upasna, if they go to your website, they'll see a whole bunch of red and green lines, all stripes all over. So I mean, one of the whiz-bang things is how can you tell whether metabolism has changed, whether genes are on and off? You might just tell them a little bit about this kind of new whiz-bang technology that allows you to do that.
Sure, yeah. So we do, what is known as RNA sequencing. So over the last, I would say 2 decades now, the technology has developed. So we have that DNA code, that ATGCs, long stretches. In the RNA, we have AUGCs. And there's, of course, thousands of different RNAs in the cell. One thing you can do is look for how the level of that RNA has changed one at a time. Or by using this methods that we use known as RNA sequencing, we can sequence all the RNA from the cells, from all this RNA from a tissue. And without going too much into detail, what I would say is basically what we do is we get the cells, we get RNA out from the cells. Then by a method, we can put some tags on the RNA. And for those tags, we know what those tags are, because the RNA, we don't know, we're just getting everything out. We don't know what is in there. So we put these identifiers, you can say molecular identifiers on both ends of the RNA. So we can hold on to them in a machine. And then we put them in a machine, and then machine can actually read which base is there. So it can say, now this is A, U, C, C, G, U, so on and so forth. So it can start reading the sequence of all of the RNA in there. So then we can take a cell sperm coming from a normal mouse and sperm coming from a low protein mouse. And then we say, okay, so all these RNA are here, all these are here too. Oh, these 2 RNA are more high in this low protein when we're getting the sequence. And that's how we can start picking up which changes are there. Now, the data can be represented, as Barry said, on our website, we have these green and red. Because we're working with thousands of molecules at a time, how do you show everything in one go? So then we say genes or RNA, which is up, they will all be shown in green. All those which are down will be all shown in red. And then you make this kind of matrix plot, very sci-fi kind, but it's actually just gene expression shown in those lines there.
Right. Point you're looking at, you sequence millions.
Yeah, millions of, I mean, there are thousands of RNA and there are many copies. So it's, in the end, we're looking at millions of reads of these. Yeah,
it's pretty amazing. Okay. Other questions for Upasna? Okay.
Yeah.
I got one down here under Sunshine Gibbs.
This reminds me of, am I unmuted? Yeah, this reminds me of the 30s when, it makes me laugh, when the Russian scientists who had no data said, we pass on the changes that happen in our life to our children.
Yeah.
Or, I think he was using lizards. I don't know what he was using.
Right, Lysenko, right, yeah. Okay.
Yeah, I think there is so much history to all of this. And many times it's been, some call it the, inheritance of acquired characters, Lamarckian inheritance. There are so many throughout history, there have been times when somebody says, oh, this is actually happening, and then the whole world will say, okay, no, there's no evidence, or unfortunately, some scientists have even committed suicide because they were proved wrong, because they thought. So if you go into the history of the whole field, it's really, yeah, sometimes you wonder, like, am I on the right field? But I think now it's getting, we have the tools now to do this, right? So for instance, the major, so August Weismann was the one guy scientist who really was the major opponent of inheritance of acquired characters, Lamarckian inheritance, because he said that there's no communication between somatic cells and germ cells, right? So as we said, we need to pass that information from somatic cells to germ cells because our body, like heart, liver, brain, all of this is exposed to the environment. And to prove that, he did an experiment, which now I think about it doesn't make sense. So what he did, he took mice, he would cut their tails and say, okay, let's see if their pups now have shorter tails. He kept doing this for generations. How many mice he killed, who knows? And he said, okay, see, so they're always coming up with a normal tail. So I disprove this. Now, if you think about it, this could be, the signal is not that you have to grow a tail. Maybe they are faster at running away from their predator, right? They can just maybe run faster. Second, he was only looking at tail. If he could do sequencing at that time, where he can look at these millions of molecules, maybe he could have found something different in those, who knows? So with the technology getting better, we I think, have a much better handle, although we still need much more new technology to do other experiments we can. But I think it's getting better because of more technology to ask these questions, looking at one phenotype of the tail versus looking at all 25,000 genes.
Thank you.
So, Upasna, so in your model where you have these small RNAs and they're in the developing egg and developing embryo, clearly those molecules are going to be degraded rather soon. So what's your idea of what's the next signal that maintains this change in the gene expression pattern?
Right, yeah, because RNA, we don't have right now any known mechanism in the embryo to keep copying these small RNAs and making more of them. So with first few divisions, we're going to dilute it out. So how do we, that early change would result in that phenotypic change we see later on. So I think this potentially, it could be some early gene expression change, which then starts a cascade of gene expression changes, which would result in, say, an altered phenotype. The genes that are changing, and this is now I'm getting more high on the scientific side, but the genes which change during early development that we picked up in these embryos, these are all required for maintaining the totipotent stage of the embryo. So what that means is it affects actually how placentation takes place, how well the placenta is formed. So there are studies which have shown that if the placenta is not properly formed, then when you grow up, you could have metabolic disorders. So placenta plays a very important role. So the genes that we identified are involved in that the development of placenta versus the embryo. So potentially these early gene expression changes have a downstream effect, is basically a secondary effect of the metabolic changes we see. In terms of, and this is kind of going back to any long lasting, let's say if transgenerationally this is passed to the next generation, then I would imagine that these early small RNAs potentially can change maybe the other epigenetic marks. So histone marks and methylation, which are more stable and could be passed down to the next generation. So this is kind of right now my thinking about how this could lead to long lasting effects.
Okay. All right. Well, I don't know if we have any last questions here. Well, that was very well explained, Upasna. It's a very complex issue. And I think you did an excellent job with it. All right. Well, thank you. Well, all right. So I hope to see you all next Saturday. Saturday gives you 2 days to do metabolic changes on everything you consumed on Thursday.
Yeah, I mean, you can reprogram your epigenome, remember.
Yeah. All right. We'll talk about how all of that food gets converted the energy, and what does that mean anyway?
Great. Thank you, everyone. Thanks, Barry.
Okay, thank you. All right, bye, everyone.
Pretty sure.
Yes. Yeah. Okay, there's not going to be any questions here.